have identified UBA1 as a novel human MK4 synthetic enzyme that catalyzes the conversion of menadione MD to menaquinone 4.
Oh, sorry. It's okay. To menaquinone 4, MK4, by the substitution of a geranil side chain at the 3 position of 2-methyl naphthoquinone ring. Oh, I can see it. Geranil pyrophosphate and farnesyl pyrophosphate and geranil pyrophosphate. All these isoprenoids derived from cellular mevalonate pathway can be substrates for UBA1 to generate MK2, MK3, and MK4 in a cell-free system using human recombinant UBA1. But when we treated mice or human cells with MDD8, neither MK2D7 nor MK3D7 was found, but only MK4D7 was detected, indicating that UBA1 is the enzyme responsible for the conversion of MD to MK4 using geranil pyrophosphate, showing the highest value of Vmax Km among the isoprenoids. Dr. Mugoni et al. Recently reported that UBA1 is a non-mitochondrial CoQ10 synthetic enzyme with specific cardiovascular protective function via modulation of eNOS activity, and that loss of UBA1 induced cardiovascular failure in zebrafish embryos by increasing oxidative stress. Supplementation with CoQ10 but not MK4 was able to rescue the vascular phenotype. In contrast, Dr. Hegarty et al. Reported that UBA1 generated MK4 but not CoQ10 rescued the zebrafish UBA1 vascular mutant, which displays cranial vascular hemorrhages because of vascular degeneration. Furthermore, Dr. Boss et al. Earlier also reported that Drosophila UBA1 hex is a modifier of PINK1, a gene mutated in Parkinson's disease with a defect of mitochondrial function, and that MK4 but not CoQ10 rescued the UBA1 hex mutant phenotype. Therefore, it remains uncertain whether UBA1 in zebrafish and Drosophila is able to synthesize MK4 like humans and mice. But it is obvious that mutations in UBA1 gene lead to severe or lethal functional failure in these species. The aims of a study were to examine biosynthesis of menadione and CoQ910 in zebrafish, whether UBA1 overexpressed neuro-2A cells derived from mouse neuroblastoma increase cellular concentrations of MK4 and CoQ910, and the biosynthesis of MK4 and CoQ910 in UBA1 knockout mice. Before showing our result, I would like to introduce briefly about CoQ910 biosynthesis in vertebrates. 4-HBA derived from tyrosine and polyprenyl pyrophosphate derived from the mevalonate pathway bind together to form a polyprenyl 4-HB and then metabolize to CoQ910. CoQ2 is a key enzyme in the CoQ910 biosynthesis and is known to involve in the formation of 3-polyprenyl 4-HB. Therefore, we first developed a method for simultaneously measuring carbon-13 polyprenil 4-HB and carbon-13 labeled CoQ9 and 10 using LC-MS-MS. We examined the biosynthesis of MK4, D7, and carbon-13 CoQ9-10 in zebrafish fed the diet containing MD-D8 and carbon-13 4-HB. As you can see in the left panels, whole extract from the zebrafish contained not only phylloquinone but also menaquinone 4, D7, as well as CoQ9, 10 endogenously. And it was found that zebrafish were able to generate MK4D7 as well as carbon-13 CoQ9, 10. Next, to examine whether UBA-D1 involves in the biosynthesis of CoQ9, 10 directly, we overexpressed CoQ2 or UBA-D1 in neuro-2A cells with or without suppression of endogenous CoQ2 expression. As you can see here, cellular concentration of MK4D7 increased significantly by transfection with UBA-D1 expression vector, but not CoQ2 expression vector. And carbon-13 solanesyl 4-HB and carbon-13 CoQ9 increased significantly by transfection with CoQ2 expression vector, as well as, but not UBA-D1 expression vector. We also obtained similar result from the same samples, showing the significant increase of carbon-13 decaprenil 4-HB and carbon-13 CoQ10 in neuro-2A cells transfected with CoQ2 expression vector, but not UBA-D1 expression vector. These results suggest that UBA-D1 converts MD to MK4, but it seems unlikely UBA-D1 catalyzes the formation of 3-polyprenil 4-HB and biosynthesis of CoQ9-10 in mouse neuro-2A cells. To examine the biological role of UBA-D1 in mice, we attempted to generate UBA-D1 KO mice. Mice UBA-D1 contains 2 exons. To disrupt UBA-D1 gene, the targeting vector was designed to flank exon 1 with 2 LOXP sequence, and the frame shift was generated by excision with recombinase. UBA-D1 heteromice were viable and fertile in comparison to control littermates. They did not exhibit any overt phenotype and had similar body size and weight. Male and female UBA-D1 heteromice were intercrossed to obtain UBA-D1 homomice. However, no UBA-D1 homopup was identified among over 150 analyzed offspring, suggesting that the disruption of UBA-D1 leads to embryonic lethality. In contrast, UBA-D1 wild and heteromice were obtained at the Mendelian frequency. To examine whether MK4 or CoQ10 treatment rescues the embryonic lethality of UBA-D1 homomice, either MK4 or CoQ10 at a dose of 10 micromole per kilogram per day or 2 micromole per kilogram per day was orally administered to UBA-D1 hetero pregnant mice from 1 day before crossing and throughout pregnancy. As a result, approximately 1.8% of UBA-D1 homo embryos were survived by E15.5, and approximately 12.5% survived by day 1 in MK4-treated UBA-D1 heteropregnant mice. In CoQ10 treated UBA-D1 heteropregnant mice, approximately 2.2% of the UBA-D1 homo embryos were survived by E15.5, and approximately 1.8% were survived by day 1. To assess the time of death in utero, in vitro fertilization (IVF) experiment was performed using sperm and eggs obtained from male and female UBA-D1 heteromice, respectively, and the embryos in utero of pseudo-pregnant mice were dissected and genotyped at several gestation times from E3.5 to E10.5. At E3.5, UBA-D1 homoblastocytes were microscopically indistinguishable from UBA-D1 wild and heteroblastocytes. However, at E7.5, both UBA-D1 wild and UBA-D1 heteroembryos showed elongation of the egg cylinder, which developed to a primitive stick with the formation of mesoderm between ectoderm and endoderm. In contrast, UBA-D1 homo embryos were consistently smaller than their retamates and exhibited no signs of primitive stick formation. The constriction marking the extra-embryonic ectoderm border was absent, and mesoderm had not formed. At E10.5, UBA-D1 homo embryos were not found at all, similar to the results observed in the intercross of male and female UBA-D1 heteromice. And UBA-D1 homo embryos at E15.5 from the UBA-D1 heteromice orally administered CoQ10 throughout pregnancy were indistinguishable from its UBA-D1 wild and heterolitamates. A willing UBA-D1 homo mouse on day 1 from the UBA-D1 heteromice orally administered CoQ10 throughout pregnancy died immediately after birth. The cause of death was not identified. In this UBA-D1 homo mouse, we couldn't find any corneal and hemorrhagic abnormalities that had been observed in SCD patients or UBA-D1 mutated zebrafish. The size and morphological features of UBA-D1 homo embryo at E15.5 and E17.5 from the UBA-D1 heteromice orally administered MK4 throughout pregnancy did not differ from those of their UBA-D1 wild and heterolitamates. These results suggest that either MK4 or CoQ10 treatment would help to rescue the embryonic lethality of the UBA-D1 homo mice, although their contribution might be small and limited. UBA-D1 homo mice were embryonic lethal for alternative confirmation of the successful ablation of UBA-D1 gene. We established ES cells from fertilized ova of UBA-D1 heteromice. UBA-D1 homo ES cells exhibited neither UBA-D1 messenger RNA and protein expression nor MK4D7 synthesis, whereas they exhibited CoQ2 messenger RNA expression and CoQ9 synthesis activity, similar to those of UBA-D1 wild and hetero ES cells. These data exclude the presence of an additional biosynthesis pathway of MK4 and indicates that UBAD1 is the sole MK4 biosynthesis synthetic enzyme in embryonic development of mice. We further examined the phenotype of 28 old UBAD1 wild and hetero mice. As expected, significant reductions of UBAD1 messenger RNA, protein expression, and MK4 biosynthetic activity in the cerebellum of the UBAD1 hetero mice were observed compared to the wild-type mice. With respect to the tissue concentrations of phylloquinone and its epoxide, there was no significant difference between the UBAD1 wild and hetero mice. In contrast, the tissue concentrations of MK4 in UBAD1 hetero mice were approximately half those of the wild-type mice. The concentrations of CoQ9 and CoQ10 in these tissues of UBAD1 hetero mice were found likely to be similar to those of UBAD1 wild-type mice. Taken together, these results strongly suggest that UBAD1 is the enzyme responsible for the synthesis of MK4, but seems unlikely for the synthesis of CoQ9 and CoQ10 in mice. In blood chemical analysis, values of total cholesterol, free cholesterol, and HDL cholesterol in UBAD1 hetero mice were significantly higher than those in UBAD1 wild-type mice, although values of LDL cholesterol and triglyceride were not significantly different between the two groups. These results suggest that UBAD1 deficiency may affect cholesterol metabolism in mice, similarly in human STD patients. Our study showed for the first time that UBAD1 is the sole enzyme responsible for the biosynthesis of MK4 in the tissues of mice, and the complete ablation of the UBAD1 gene leads to embryonic lethality, as rescued only partially by oral supplementation with MK4 or CoQ10. UBAD1 may play a critical role in embryonic development through the biosynthesis of MK4. But an alternative UBAD1 MK4 independent pathway may be possible to involve in the embryonic development of mice. Given that systemic UBAD1 knockout mice uniformly die between E7.5 to E10.5, the elucidation of the physiological and pathological rules of UBAD1 will require the generation of mice exhibiting tissue-specific deficiency of UBAD1 without embryonic and postnatal lethality. Thank you very much for your attention.
Thank you. We have some time for questions.
Okay.
What do you think would be the function of vitamin K during early stage of development at this point?
We have no explanation about this, but we imagine maybe it has antioxidant activity, antioxidation response. So we imagined it, we guess it, we believe it. Yes.
But how that would work?
Yes. My colleague, Dr. Nakagawa, presented earlier. We have done many experiments focusing on the antioxidant activity of MK4 in brain tissues, as well as neuron and astrocyte isolated. So far we have got positive results to explain the antioxidant activity is very important for the neuronal cells survival.
Very nice presentation,
Thank you.
Toshio. I really liked it. So you had intermediate levels of MK4 in the heterozygote,
Yes.
but yet this is due to enzyme function. So isn't this a little bit unusual to have, you know, an enzyme showing an intermediate level?
Yeah.
This is more common, like for instance, like LDL receptor deficiency. But when you have an enzyme, that this would be such so rate limiting in the heterozygous state. Can you comment on that?
Yes, we are lucky to find the almost half, but it's I think so. It's not so usual, but as you know, homozygote of the UBA1 does not exist in the humans. Maybe MK4 synthesis is strictly regulated. I showed previously each tissue has its own levels of MK4, and we injected or we administered warfarin or other inhibitors of this enzyme. Dramatically, the levels of the MK4 tissue levels dramatically dropped to the almost 1/10, and then increased to the original levels. So we have no scientific data to explain this, but I think the concentration, cellular concentration of MK4 is somehow regulated by this enzyme. That's why this enzyme is very important to synthesize MK4. But maybe via some regulations.
Okay, thank you.